We're going to be talking about DNA synthesis by that high school tutor. So, DNA synthesis is complicated, as most things are. So, I'm going to try and explain this to you in the easiest way possible. So, in our last video, we talked about the two phases of the cell cycle. So, let's do some review. As I said before, there are two phases, interphase and M phase. In this video, we're mostly going to be talking about what happens in interphase, specifically S phase. Here's some review if you wanted to see. And let's move on. So I have a glossary for you guys because some of the terms in here you might not know. So let's review. Nucleotides are the basic building blocks of nucleic acids, aka DNA and RNA. They consist of adenine, thymine, cytosine, and guanine, or the famous acronym ATGC. Adenine goes with thymine and cytosine goes with guanine, and you'll see that in a few minutes. Parental DNA or original DNA molecule, self-explanatory, and the daughter molecules are the result of DNA synthesis of the parent DNA. Self-explanatory, you'll see what happens with them in a few slides. So, what happens in DNA synthesis? So DNA synthesis is a process where DNA is copied in a cell before division, and specifically in the nucleus of the cell. This DNA is all coiled into a chromosome originally, but it becomes X-shaped chromosomes after this stage, not like we mentioned in our last video. So, we have 46 of these X-shaped chromosomes in our body, and they get passed on to our daughter cells as well. So, just for some reference. So, this is our original strand. That's where you see ATGC, adenine, thymine, guanine, cytosine. So, this is what we call the original strand of DNA, or also known as the parental molecule of DNA. Now, this gets split apart, and new things are added, which are called nucleotides. And these are, again, the parental strands of DNA. After that, you have two new identical daughter molecules, or two identical DNA strands. This whole process is called semi-conservative replication because the original strands here become templates for the new strands. If that's too confusing, semi-conservative basically means half kept. So half of this parental molecule is kept in one DNA, and another half is kept in another DNA strand. There we go. So what are the enzymes? There are four main enzymes in DNA synthesis: helicase, primase, polymerase, and ligase. They are very important in DNA synthesis, and without them, there will not be any DNA synthesis. So what do each of these things do? They're all involved in the steps of DNA synthesis, aka replication. So DNA separating begins with the enzyme helicase, ding, ding, ding. So this is a picture that shows you what helicase does. It's not the accurate shape of helicase, but it shows you the gist of what it does. It separates these two strands, and to prevent them from coming back together again, there are proteins that prevent it from doing that. Okay. So the next step in DNA replication is the enzyme primase, ding, 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 lays down an RNA primer, primase primer. There you go. This is a primer that acts like a signal so that the next enzyme in DNA replication, polymerase, can do its job. So let's see a picture. There we have helicase, our first enzyme in DNA replication, and we have primase right here. Now, primase lays down this RNA primer right here, which basically shows that shows to polymerase, like, okay, this is supposed to go with this, and this is supposed to go with this. That's basically its job. Now, we don't talk about topoisomerase. And I'm really bad at pronouncing this, but basically it stops from making this coil really much, because whenever you separate something that's already coiled, that helicase does, it tends to get more coiled. It's just so this enzyme prevents that from happening. Okay. Next step. The next step is the enzyme polymerase starts adding complementary nucleotides, ATGC, to each of the original DNA strands. This is really complicated, so I'm going to explain it to you in a simple way. So, 5' and 3' are the structure of how DNA is formed, and it has to do with the deoxyribose sugar that are attached to each of these bases here. We call ATGC nitrogenous bases. So you see here 5' is here and 5' is here. That's why this end is called 5' and this end is called 5'. And you can see this strand of DNA is actually flipped over. That's why they have opposite ends. And if you want to learn more about this, to really understand what's going on in polymerase, there are some links in the description that may help you. So, this is really important because DNA, it grows from 5' to 3' end. It grows, this one goes this way and this one goes this way. But polymerase, the enzyme, builds this way, 3' to 5', which is the opposite. And I'll show you why this is super significant. So here's a diagram. We see polymerase, the triangle here and here. And we see that there's a gray line and parts of a gray line here. This gray line is called the daughter strand, or the new strand that's being synthesized. This enzyme, polymerase, is adding those ATGC strands. Complementary means like A goes with T and G goes with C. So it's putting those strands that go with it. So why is this broken up? Well, as we said before, polymerase builds 3 to 5. And since this strand is going from 3 to 5, it's able to build continuously this way. But on this side of the DNA, since we said before, the original strand of DNA is flipped, 3', 3', 5', 5'. It actually lags behind. So whenever it starts in something, it has to jump back to start a new line. So it goes here, up, we gotta go down. Up, we gotta go down. Because it doesn't have time while helicase is making new openings, polymerase has to keep jumping back to keep making builds. This is why we call these certain strands names. The leading strand is this gray continuous line because it's a straight line. And this is called the lagging strand because polymerase is lagging behind. See these little strips? Now, these synthesized pieces of DNA are called Okazaki fragments, and they were named after the Japanese scientist with the last name Okazaki because he found out what these were. Synthesize is just another word for build, just to make everything simpler. Now, if you want some more detail on how this polymerase lags in detail, I left a video in the link, so check that out if you're interested. So, the last step in DNA replication is the enzyme ligase, which ties together the small pieces, the Okazaki fragments, in the lagging strand, and they tie them together. So here's a diagram. You see these Okazaki fragments here. this DNA ligase acts like a glue and makes these two so that there's no gap in between here. As you can see, the overall direction of replication is still going this way. Helicase, imaginary helicase, is still cutting its way through the DNA, and polymerase is still adding those bases. All of these things work together to split DNA, but it would take a very long time for only one spot where this is happening. DNA replication just doesn't happen in one spot of DNA. It happens in many, many spots. That's why it gets copied very fast. Just to put that in picture. So that's the end of DNA replication. I have some questions for you guys to test your knowledge. So pause this video and see if you can answer some of these questions. Okay, here are the answers. If any of you guys have a question, leave it in the comments and I'll get back to you on them. Thank you so much for watching and have a great day. I hope you learned something new today. Bye!